So we're proud to say that since 2001, we've enrolled families from 49 states within the U.S., from 28 countries and 6 continents. And, oh, you can't see it? Oops, sorry. This is a picture of the world. All you see are stars. It looks really nice on my screen. And these are some of the locations and geographic areas where some of our families come from. And every family that we enroll into our study is helpful and provides a great deal to our research. So to date, we've enrolled 385 individuals with CDH. We've enrolled 750 family members without CDH, and 35 of our families are considered multiplex, or there's more than one affected relative. And we've also enrolled 8 sets of twin pairs which are identical, and they're discordant for CDH, meaning that one twin has the CDH and the other one doesn't. And since identical twins are thought to have the same DNA at the nucleotide level, the letter level basically, GCTA, it's interesting for us because it allows us to look at different types of genetic variation. So scientists estimate that there is about 30,000 genes within the human genome. And unfortunately, we don't fully understand which genes are required for proper diaphragm development, and which of those genes, when they're not working right, lead to CDH. So in our lab, we use a broad range of interdependent genetic approaches to try to uncover genes that are causative or that lead to CDH when they're not working right. So how do we learn about CDH? We use a variety of different methods, and some of them are genetically manipulated animal models. So different labs use different models. In our laboratory, we use the mouse, but you can use rodents and all sorts of models. We learn about CDH through CDH plus associated anomalies, like Mauro was just talking about, so that when you have abnormalities that are more commonly occurring with CDH, like limb abnormalities or heart abnormalities, maybe the genes that are responsible for the growth of the limb or for development of the heart, maybe those genes also interact or play a role in diaphragm development. So we look at those genes as well to see if they're not working properly and perhaps they play a role in the diaphragm. We look at regulatory mechanisms of diaphragm embryogenesis or early diaphragm formation, because we want to say, okay, well, how is the diaphragm formed correctly? Maybe it's not forming correctly, and that's why those genes are being perturbed. And we also look at CDH chromosome hotspot regions, because those genes contained within those regions, or those that are deleted or rearranged, they might be causative for the defect like you guys learned about yesterday. And they are causative. So how do we find our candidate genes? We use a number of different cytogenetic and genomics, and we use all different kinds of approaches in our lab to try to narrow down and find candidate genes. And some of the ways we do this are by CDH analysis, which is copy number analysis. We perform genome-wide SNP analysis. We do expression studies on different tissues. We do pathway sequencing, and then we do bioinformatics and computational biology to try to weed through our data and mine it. And we do these studies on our CDH cohort. So in the affected individuals, we take DNA, and then we do all these different kinds of studies, depending on the child as well. And we want to understand CDH in general, but we also look at CDH within each family, because everybody's different, and everybody's classified very well by the geneticists, so we know we're not going to group certain things with others. So what I'm going to do is first focus on how we learn about CDH using animal models, since last year, more of the technical stuff about different techniques we use. This time I'll talk about how we learn through animal models, and then I'll go ahead and talk about how we find candidate genes through the use of bioinformatics and computational biology. Computational analysis. So mice or rodents in general actually do teach us a lot about human development. And that's because, sorry, that's a mouse. You can't really see him eating cheese. It's because they're easy to analyze because you can manipulate their genomes, and you can analyze their genomes pretty easily. And they also have a resemblance to human physiology and human genetics. So in our lab, as I said, we use the mouse, but this is a picture of, or a drawing of the human diaphragm that's fully formed and formed properly. And you can see that this is the muscularized region of the diaphragm. And you're actually looking from below, so you're within the abdominal cavity. So, and this is the amuscular central tendon region. So those are key components. And since the diaphragm forms around the 4th week of human gestation and is thought to be complete around the 12th week, but it's sort of arranged and different people say different things. It's very early, so it's in the 1st trimester. So it's really difficult to study human development of the diaphragm. And that's probably one of the reasons why there's not much out there. If you try to do literature researches on diaphragm development, there's a few people that do it, and they do it, and that's what they do, but there's not much beyond that. I think that's why it's been so tough. So we use the mouse as a model. This is actually a picture of a rat diaphragm, but it's very similar to the mouse. It was just a decent comparison. So again, you can see the similarities. These are the muscularized regions, and this is the central tendon region. It's not muscular. So what I just showed you was a mature diaphragm, but the time period that we're interested in studying diaphragm development is a lot earlier. So we're interested at around 5 weeks of human gestation. So this is actually a human embryo taken from a embryology book. And you can see that this is the head, the brain, the different parts of the brain. We have our spinal cord. We have our limbs that are going to be growing, our legs and our arms. But this is the region where, sorry I forgot my laser pointer, where the diaphragm is going to be formed. And it's below the region up where the heart is forming and where the lung buds are, or where they're forming. So because we don't study the human this early in development, we use the mouse. And just to give you a comparison of how they're similar, this is a mouse embryo at day 11.5, which is the time period that we often study diaphragm development. And we use this, and many people do. It's when just right before the diaphragm is, we have these, I'll show you on the next slide, but it's the primordial diaphragm is just about to form. And you can see the head, here's the spine, we have our heart, we have our lungs forming, and the diaphragm is going to be over here. It's going to form this way, for the early diaphragm. So what we do to study these mouse embryos at embryonic day 11.5 is we take the embryos and we put them into a wax, it's called paraffin. We put it into the wax and we embed it and we actually create sections to get the correct tissue. So this is sort of an example in the corner here. So let's say this is in a piece of wax and we section with a special, and we get the embryo. We get the developing diaphragm right here at this level. And to give you, this is the nose here. Here's the nose. Here's our heart. Here's our heart. Then we have our lungs and our spinal cord. And here's our limbs. So the area of interest for the primordial diaphragm, which we call the pluriperitoneal folds, are right here. So this is what we study, believe it or not. These regions here are how we study diaphragm development, because these tissues are going to give rise to the diaphragm. And actually, if the pluriperitoneal folds, or these early diaphragm, doesn't form properly, you'll end up with a congenital diaphragmatic hernia. You'll end up with a CDH. So this is an example of another section. Sorry, it's so bright. You can't really see it. But this is another section, the same orientation. This I just drew in blue where this pluriperitoneal fold is, and it's nice. And you can see here, it's missing. And this later translates into a hole in the diaphragm. And that's a fully formed, mature diaphragm. This is all mouse, or this is actually rat. So what we end up doing then is we can say, okay, well, obviously there's a problem going on early, so we can study the pluriperitoneal folds, but we also want to study other developmental processes that have to work together to create the diaphragm. And there's many that are going on. And I'm just going to give you an example.